Welcome to today's session of food technology, under which you will be learning about minerals. Let us start with the introduction. In continuation with the first part, let us learn about microminerals in this unit. In earlier unit, you learnt about the definition and classification of minerals. You learnt about the functions, deficiency and excess of macrominerals and ultra trace minerals. This chapter describes functions, deficiency and toxicity of microminerals. A precise definition for the essential of microminerals has not been established. The term micro, when applied to minerals, can be defined as minerals that can make up to less than 0.01% of total body weight. An element is considered essential if a dietary deficiency of that element consistently results in a suboptimal biological function that is preventable or reversible by physiological amount of element. The abnormalities induced by deficiencies are always accompanied by a specific biochemical changes. Each essential micromineral is necessary for one or more functions in the body. Whenever the intake or body concentration is too low or too high, function is impaired and death can result. The body's content of the microminerals ranges from less than 1 mg to approximately 4 gram. Through study of this unit, you will be able to understand functions and deficiency and excess of the following microminerals, which are iron, zinc, copper, fluoride, manganese, chromium, iodine, molybdenum and selenium. Now, let us start with the first micromineral, iron. The human body contains approximately 2 to 4 gram iron or approximately 38 mg iron per kg body weight for women and around 50 mg iron per kg body weight for men. Iron exists in a complex form in our body. It is present as iron porphyrin compounds, as enzymes and as transport and storage forms. In iron porphyrin compounds, it contains hemoglobin and myoglobin. Hemoglobin is present in RBC, which is around 65%, and in myoglobin in muscles, which will be around 10%. And in enzymes, it is 1 to 5%, which contains peroxidases, succinase dehydrogenase and cytochrome oxidase. In transport and storage forms, it is around 20%, which are transferrin and ferritin. The total amount of iron found in a person not only is related to body weight, but also is influenced by other physiological conditions including age, gender, pregnancy and state of growth. Now, let us see the functions. The chief functions of iron in the body are: iron forms a part of the protein hemoglobin, which carries oxygen to different parts of the body. It forms a part of myoglobin in muscles, which makes oxygen available for muscle contraction. Iron is necessary for the utilization of energy as part of the cell's metabolic machinery, and as part of enzymes, iron catalyzes many important reactions in the body. Examples are conversion of beta carotene to active form of vitamin A, synthesis of carnitine, purines and collagen, and in detoxification of drugs in the liver. Now, let us see the deficiency of iron. Iron deficiency occurs most often due to inadequate iron intake. Iron intake is frequently inadequate in 4 population groups. They are infants and young children, adolescents, females during childbearing years and pregnant women. In infants and young children, and the age group will be 6 months to about 4 years, because of the low iron content of milk and other preferred foods, rapid growth rate and insufficient body reserves of iron to meet needs beyond about 6 months. In adolescent groups, in their early growth spurt, because of rapid growth and the needs of expanding red blood cell mass. Females during childbearing years because of menstrual iron losses. In pregnant women because of their expanding blood volume, the demands of fetus and placenta and blood losses to be incurred in childbirth. In addition, many non-pregnant females during childbearing years fall short of the recommended dietary allowances for iron because of restricted energy intake and inadequate consumption of iron rich foods. Dietary iron deficiency leads to nutritional anemia. Nutritional anemia is defined as the condition that results from the inability of the erythropoietic tissue to maintain a normal hemoglobin concentration. Anemia occurs when the hemoglobin level falls below 12 gram per dl in adult man and women. During pregnancy, hemoglobin level below 11 gram per dl is termed anemia. The major causes of anemia in India is because of iron and folic acid deficiency. Nutritional anemia is manifested as reduced hemoglobin level, defects in the structure, function of the epithelial tissues, paleness of skin and inside of the lower eyelid is pale pink, fingernails becoming thin and flat, and eventually koilinochia develops, which is spoon shaped nails. Progressive untreated anemia results in cardiovascular and respiratory changes leading to cardiac failure. The general symptoms include lassitude, fatigue, breathlessness on exertion, palpitations, dizziness, sleeplessness, dimness of vision, and increased susceptibility to infection. Symptoms of iron deficiency mostly demonstrated in children include pallor, listlessness, behavioral disturbances, impaired performance in some cognitive tasks, some irreversible impairment of learning ability, and short attention span. Now, let us see the excess of iron. Accidental iron overload has been observed in young children following excessive ingestion of iron pills or vitamin or mineral pills. In most people with hematochromatosis, iron absorption generally continues despite high iron stores. The absorbed iron is progressively deposited within joints and tissues, especially the liver, heart and pancreas, causing extensive organ damage and ultimately organ failure. Now, let us see the next micromineral, zinc. The human body contains approximately 1.5 to 2.5 gram of zinc. Zinc is found in all organs and tissues and in body fluids. Largest stores of zinc is present in the bones. Zinc forms a constituent of the blood. Zinc is an important element performing a range of function in the body as it is a cofactor for a number of enzymes. Functions: Zinc function in association with more than 300 different enzymes. It participates in reactions involving either synthesis or degradation of major metabolites like carbohydrates, lipids, proteins and nucleic acids. It plays important structural role in brain cells. Zinc is involved in the stabilization of protein and nucleic acid structure as well as in transport processes, immune function and expression of genetic information. It plays a major role in synthesis of DNA and proteins and a constituent of the hormone insulin. Zinc appears in the crystalline structure of bone in bone enzymes. It is thought to be needed for adequate osteoblastic activity, formation of bone enzymes such as alkaline phosphatase and calcification. The deficiency: some population groups, especially the elderly and vegetarians, have found to consume less than adequate amounts of zinc. Conditions associated with an increased need for intake include alcoholism, chronic illness, stress, trauma, surgery and malabsorption. Signs and symptoms of zinc deficiency are growth retardation, skeletal abnormalities, defective collagen synthesis or cross-linking, poor wound healing, dermatitis, delayed sexual maturation in children, hypogeusia, alopecia, impaired immune function and impaired protein synthesis. Excessive intake of zinc can cause toxicity. An acute toxicity with 1 to 2 gram zinc sulphate, as zinc will be 225 to 450 mg, can produce a metallic taste, nausea, vomiting, epigastric pain, abdominal cramps and bloody diarrhea. The third micromineral is copper. The adult body contains approximately about 80 mg copper. Concentrations of copper are highest in the liver, brain, heart and kidney. Muscle contains a low level of copper, but because of its large mass, skeletal muscle contains about 40% of all the copper in the body. Functions: copper is a component of many enzymes and symptoms of copper deficiency are attributable to enzyme failures. Copper in ceruloplasmin has a well documented role in oxidizing iron before it is transported in the plasma. Lysyl oxidase, a copper containing enzyme, is essential in the lysine derived cross linking of collagen and elastin. Copper has roles in mitochondrial energy production. As part of copper containing enzyme such as superoxide dismutase, copper protects against oxidants and free radicals and promotes the synthesis of melanin and catecholamines. Clinical manifestation associated with copper deficiency are hypochromic anemia, leukopenia, hypopigmentation or depigmentation of skin and hair, impaired immune function, bone abnormalities, cardiovascular and pulmonary dysfunction. The likelihood of copper deficiency increases in persons consuming excessive amounts of zinc or antacids, as well as in the persons with conditions that promote increased loss of copper from the body, as occurs with nephrosis or gastrointestinal malabsorptive disorders such as celiac disease, tropical sprue, and inflammatory bowel diseases. Excess copper toxicity is fairly rare. A tolerable upper level for copper is set at 10 mg per day. Copper intake of 6 to 4 mg has resulted in epigastric pain, nausea, vomiting and diarrhea. Other symptoms of toxicity include hematuria, liver damaging resulting in jaundice and kidney damage resulting in oliguria or anuria. Wilson's disease, a genetic disorder characterized by copper toxicity which results from mutations in the gene coding. In Wilson's disease, copper accumulates in organs resulting in disturbed function of organs, especially the liver, kidneys and brain. Kayser-Fleischer rings caused by copper deposition also are visible in the cornea. Now, let us see the fourth micromineral, which is fluoride. Fluoride is found in natural element in nearly all drinking water and soil and in the human body in trace amounts. Fluoride is not considered an essential nutrient, but it is clearly recognized as important for the health of bones and teeth. Functions: fluoride is essential for tooth enamel. Fluoride incorporation into enamel produces more stable apatite crystals. Fluoride also acts an antibacterial agent in the oral cavity, serving as an enzyme inhibitor. Deficiency: fluoride deficiency in test animals have been reported to result in curtailed growth, infertility and anemia. In humans, an optimal level of fluoride helps to reduce the incidence of dental caries and perhaps also to maintain the integrity of skeletal tissues. Excess chronic toxicity of fluoride, called fluorosis, is characterized by changes in bone, kidney, nerve and muscle function. Dental fluorosis or mottling of teeth has been observed in children receiving about 2 to 8 mg fluoride per kg body weight. Acute toxicity manifests as nausea, vomiting, diarrhea, acidosis and cardiac arrhythmias. The fifth micromineral is manganese. Although widely distributed in nature, manganese occurs in only trace amounts in animal tissues. The body of a healthy 70 kg man is estimated to contain a total of 10 to 20 mg of the metal. Functions: manganese is a component of many enzymes including glutamine synthetase, pyruvate carboxylase and mitochondrial superoxide dismutase. Manganese is associated with the formation of connective and skeletal tissues, growth and reproduction and carbohydrate and lipid metabolism. Deficiency: manganese deficiency is associated with striking and diverse physiological malfunctions. Manganese deficiency generally does not develop in humans unless the mineral is deliberately eliminated from the diet. Symptoms and signs of deficiency includes nausea, vomiting, dermatitis, decreased serum manganese, decreased fecal manganese excretion, increased serum calcium, phosphorus and alkaline phosphatase, decreased growth of hair and nails, changes in hair and beard color, poor bone formation and skeletal defects, and altered carbohydrate and lipid metabolism. Excess manganese toxicity has developed in miners as a result of absorption of manganese through the respiratory tract. The excess, which accumulates in the liver and central nervous system, produces Parkinson-like symptoms. Toxicity has also been reported in patients receiving total parenteral nutrition including manganese. Symptoms include headaches and dizziness. Next micromineral is chromium. Chromium is found in air, water and soil. The chromium content of human body is estimated at 4 to 6 mg. The functions of chromium: chromium potentiates insulin action and as such influences carbohydrates, lipid and protein metabolism. Chromium may regulate the synthesis of a molecule that potentiates insulin action. Another possible role for chromium, similar to that of zinc, is in the regulation of gene expression. Next is deficiency. Chromium deficiency is seen in people who received intravenous nutrition feeding without chromium and without oral food intake. Signs and symptoms of deficiency includes weight loss, peripheral neuropathy, elevated plasma glucose concentrations or impaired glucose use and high plasma free fatty acid concentrations. Severe trauma and stress may increase the need for chromium. Chromium deficiency results in insulin resistance characterized by hyperinsulinemia, a risk factor for heart disease. Mild chromium deficiency also is a risk factor for metabolic syndrome. Metabolic syndrome increases the risk of heart disease. Next is excess. Oral supplementation of up to 1000 microgram of chromium appears to be safe. Toxicity is associated with exposure to chromium absorbed through the skin, enter the body through inhalation or be ingested. Inhalation of or direct contact with chromium may result in respiratory disease or in dermatitis and skin ulcerations. Liver damage may also occur. Ingesting chromium leads to severe acidosis, gastrointestinal hemorrhage, hepatic injury, renal failure and death. Next is iodine. Iodine is an essential micronutrient which is required for the synthesis of thyroid hormone for optimal physical growth and development of humans. The healthy human body contains about 20 mg of iodine, 70 to 80% of which is concentrated in the thyroid gland. Now, let us see the functions of iodine. Iodine is stored in the thyroid gland where it is used in the synthesis of triiodothyronine and thyroxine. Uptake of iodide ions by the thyroid cells may be inhibited by goitrogens. Goitrogens are the substances that exist naturally in foods. Thyroid hormone is degraded in target cells and the liver, and the iodine is highly conserved under normal conditions. Deficiency. Iodine deficiency in the diet causes enlargement of thyroid gland called as goiter, as well as a wide spectrum of disorders which are termed as iodine deficiency disorders. Iodine deficiency disorders includes abortion, stillbirths, low birth weight, cretinism, neonatal chemical hypothyroidism, psychomotor defects, impaired coordination, mental retardation and hypothyroidism. Goiter occurs in people staying in hilly regions where the iodine content of water and soil is comparatively less. Goiter can be treated by administration of iodine. If treatment is given in early stages, goiter can be corrected. Severe iodine deficiency in children leads to hypothyroidism resulting in retarded physical and mental growth. This condition is known as cretinism. Goitrogens are substances present in foods which cause goiter. These substances react with the iodine which is present in the food, making it unavailable for absorption. Foods like cabbage, cauliflower, radish contain goitrogens. Excessive iodine intake is reportedly occurring because of poor monitoring and higher than necessary supplementation in several countries with supplementation programs. In addition, in some countries, excessive intake occurs from overconsumptions of food naturally high in iodine. Some signs of acute iodine deficiency include burning of the mouth, throat and stomach, nausea, vomiting, diarrhea and fever. A tolerable upper intake level for iodine has been set at 1100 microgram per day. Next micromineral is molybdenum. The need for molybdenum was established in humans through the observation that a genetic deficiency of specific enzymes that require molybdenum as a cofactor resulted in severe pathology. Functions of molybdenum: the xanthine oxidase, aldehyde oxidase and sulfite oxidase, all enzymes that catalyze oxidation reduction reactions require a prosthetic group containing molybdenum. Deficiency: molybdenum deficiency has not been established in humans other than patients treated with total parenteral nutrition. Symptoms of molybdenum deficiency include mental changes and abnormalities of sulfur and purine metabolism. Next is excess. Molybdenum appears to be respectively non-toxic with intake up to 1500 microgram per day. However, symptoms such as gout have appeared in some people living in regions that contain high soil molybdenum levels and in those with occupational exposure of molybdenum. Next and the last micromineral is selenium. Selenium, a non-metal, exists in several oxidation states. Selenium is important as it has antioxidant activity. The chemistry of selenium is similar to that of sulfur. Consequently, selenium can often substitute for sulfur. The total body selenium content ranges from about 13 to 30 mg. Now, let us see the functions of selenium. Glutathione peroxidase is the only selenoprotein enzyme well studied for the biological role of selenium. Diiodinase isoenzymes that are involved in thyroid hormone metabolism are also selenium containing proteins. Apart from antioxidant protection against free radicals, selenium was found to be functional in detoxification. The deficiency. Selenium deficiency has been associated with two childhood or adolescent endemic diseases like Keschard and Keschinbeck. These diseases are found to be prevalent in certain areas where the intake of selenium is very low, which is around 7 to 11 gram per day. Poor intake of selenium is associated with increased risk of cancer or heart disease. Now, let us see about the excess of selenium. Selenium toxicity, also called as selenosis, has been observed both in minors and in people who consume excess selenium from supplements. Signs and symptoms of toxicity include nausea, vomiting, fatigue, diarrhea, hair and nail brittleness and loss, paresthesia, interference in sulfur metabolism and inhibition of protein synthesis. Daily intakes about 700 microgram per day or acute consumption of 1 to 7 mg selenium per kg per day results in toxicity in humans. Now, let us conclude this session. Micro, when applied to minerals, can be defined as minerals that make up to less than 0.01 percentage of total body weight. The body's content of the microminerals ranges from less than 1 mg to approximately 4 gram. The microminerals are iron, zinc, copper, fluoride, manganese, chromium, iodine, molybdenum and selenium. Microminerals functions in association with different enzymes. They participate in reactions involving synthesis, degradation of major metabolites like carbohydrates, lipids, proteins and nucleic acids. They play important structural role and many biochemical functions. The abnormalities induced by deficiencies are always accompanied by specific biochemical changes. These biochemical changes can be prevented or cured when the deficiency is prevented or cured. Whenever the intake of body concentration is too low or too high, function is impaired and death can result. Thank you.